How honey is made, the secret life of the hive, and the journey from nectar to gold. Greetings to all who appreciate not only taste, but also the amazing stories hidden in everyday objects. Today, we'll calmly and clearly explore the creation of one of the oldest and most perfect natural products, honey. We'll trace the journey from a drop of flower nectar to the thick aromatic syrup in a jar, learn how thousands of tiny creatures act as a single highly organized organism, and understand the complex biochemistry behind a sweetness that even centuries cannot spoil. Imagine flawless wax hexagons filled with a thick substance, ranging in color from sunny gold to deep amber, scented with meadows, forests, and blooming gardens. This is honey, a product that humanity has prized for millennia, not only for its taste, but also for its amazing properties. But how does it come about? The common belief that bees simply collect honey like we pick berries in the forest is a gross oversimplification. In fact, honey isn't collected, it's produced. It's the result of one of nature's most complex and well-oiled biochemical processes, carried out by a community of tens of thousands of insects, each a cog in a gigantic living conveyor belt. From raw material collection to final packaging, each stage has its own role, and the goal of the entire process is to ensure the survival of the bee colony. Let's take a look inside the hive and follow this journey to understand this miracle of nature we spread on our morning toast. The process of honey production begins not in the hive, but many kilometers away, in the world of flowering plants that have entered into an ancient and mutually beneficial alliance with pollinating insects. The primary raw material for honey production is nectar, a complex plant secretion. Nectaries, specialized glands of flowers, produce a sweet sap whose primary function is to attract insects that, moving between plants, ensure cross-pollination. However, from the bee's perspective, nectar is only a primary imperfect raw material. Biochemically, it is predominantly water with a content ranging from 50% to 80%, making it extremely unstable and susceptible to fermentation. The dry matter is represented primarily by sugars, the disaccharides sucrose, cane sugar, and the monosaccharides glucose and fructose. The ratio of these sugars in the nectar of a particular plant will largely determine the physical properties of the future honey, its crystallization rate and density. In addition to sugars, nectar contains vitamins, B and C, organic acids, dextrins, nitrogenous compounds, and crucially, volatile aromatic compounds and essential oils, which form the basis of the honey's unique bouquet, allowing an experienced connoisseur to determine its botanical origin, whether it's linden, buckwheat, or sunflower. Nectar is not the only source for honey production. There is a second, less romantic but no less important raw material, honeydew. This is not a plant product, but the secretions of small insects, aphids, psyllids, and scale insects that feed on plant juices. Their digestive system digests the proteins in the sap, and the excess sugars and water are excreted as sweet droplets on the leaves and shoots of trees, oak, maple, linden, and conifers. Chemically, honeydew differs significantly from nectar. It contains more oligosaccharides, melizotose, raffinose, minerals, potassium, magnesium, iron, and dextrins, but fewer simple sugars. Bees collect this substance during periods when flower nectar is unavailable, often in late summer or during dry, hot weather. Honey produced primarily from honeydew is called honeydew honey. It is dark in color, ranging from dark brown to almost black, less sweet, sometimes with a bitter or malty flavor, and has a viscous, syrupy consistency. It is highly prized in Europe for its prebiotic and mineral properties. In Russia, however, demand for lighter, sweeter floral varieties is traditionally higher. A critical issue for the effectiveness of the entire bee colony is search and exploration. A colony numbering tens of thousands of individuals must optimally distribute its labor resources. This task is performed by scout bees, experienced flyers with exceptional abilities to navigate and memorize the landscape. Having discovered an abundant source of nectar or honeydew, a scout returns to the hive and transmits information using a symbolic language unique in the animal kingdom, a dance. If the source is nearby, within 50 to 100 meters, she performs a simple circle dance, prompting other bees to search in the immediate vicinity. If the find is far away, the bee switches to a complex waggle dance. Moving along the comb, she traces a figure-8 pattern. A straight line in the middle of this figure, the belly, indicates the direction of the target relative to the position of the sun. If a bee runs up the comb, the source is located directly toward the sun. If it runs down, it's away from the sun, and the angle of deviation from the vertical precisely corresponds to the angle between the sun, the hive, and the food source. The energy of the dance and the number of repeated cycles indicate the abundance of the source and its distance. The farther away, the slower the dance. Furthermore, the scout shares a nectar sample with other bees, allowing them to memorize its taste and smell. This phenomenal communication system, studied by the science of ethology, transforms the hive not into a cluster of individual insects, but into a single superorganism, capable of making collective decisions that optimize survival, distributing foragers to the most promising areas of the landscape. Once information about a rich source is received, a well-oiled production line is set into motion, with each bee performing a strictly defined function depending on its age and physiological state. The first link is the forager bee. Armed with a long proboscis, it sucks up nectar or honeydew, which enters not the digestive stomach, but a special organ, the honey stomach. This is a separate, expandable reservoir, a kind of test tube for the bee, where the raw material is isolated from the digestive juices. The stomach holds approximately 40-70 milliliters, and to fill it, the bee must visit from several hundred to 1,000 or more flowers, flying many kilometers in the process. Once in the stomach, primary processing begins. The nectar is mixed with a secretion from the pharyngeal glands, rich in the enzyme invertase. This is the beginning of the key biochemical process of inversion. Returning to the hive, the forager takes her time storing her spoils in the cell. She finds one or more receiver bees, either hive or flightless bees, and transfers a drop of nectar to them through her proboscis in a process called trophallaxis, food exchange. This act is not simply a transfer of raw material, but a crucial technological step. The receiver bee repeatedly, over the course of 15 to 20 minutes, releases a drop onto her coiled proboscis and then draws it back into her crop. This procedure, reminiscent of pouring, serves several purposes: further enriching the nectar with enzymes from the receiver's glands, active mixing, and most importantly, evaporating water from the relatively large surface area of the drop. This is how primary condensation occurs. After this, the partially dehydrated and fermented syrup is placed in a wax cell by the receiver. However, this is not yet honey. At this stage, the humidity of the contents is still too high, around 50 to 60%. To complete the maturation process, the active ventilation system is activated. Numerous bees position themselves at the hive entrance and on the combs inside the hive, beating their wings at an incredible rate, creating a powerful directed airflow. This draft, permeating all the streets of the hive, accelerates the evaporation of moisture from the open cells. The evaporation process can take from several days to a week, depending on the humidity of the surrounding air. The bees constantly monitor the state of the syrup, moving it from one cell to another as needed to ensure uniform maturation. The final stage of production is sealing, when, thanks to enzymes and evaporation, the humidity of the cell contents drops to a critical level of 17 to 20%, and the complex sugars are mostly converted to simple ones. The bees receive a signal that the product is ready for long-term storage. They seal the cell with a breathable but tight cap made of pure wax mixed with pollen. This wax seal is a sign for the beekeeper and a guarantee for the colony that the honey is mature, possesses all the necessary properties, and will not ferment. Thus, the path from nectar to preserved honey is an example of an ideal natural technological cycle where raw materials sequentially undergo the stages of collection, enzymatic processing, concentration, and packaging with quality control built into the process at every stage. The transformation of nectar into honey is not simply a physical evaporation of water, but a complex biochemical process controlled by specific enzymes added by bees at various stages. It is this enzymatic processing that transforms the unstable plant sap into a unique product with preservative properties, capable of being stored for centuries without loss of quality. 3 key enzymes play a key role here: invertase, diastase, amylase, and glucose oxidase. The first and most important stage is sugar inversion. Nectar contains a significant amount of the complex disaccharide sucrose, common cane sugar. The enzyme invertase, secreted by the bee's pharyngeal glands, breaks down the sucrose molecule into 2 simple monosaccharides, glucose and fructose. This process is fundamental. Firstly, simple sugars are more easily digested by both bees and humans. Secondly, it prevents the potential crystallization of sucrose in the honeycomb, which would make the honey unsuitable for winter feeding. In the final product, the ratio of glucose to fructose determines its physical properties. Honey with a predominance of glucose, for example rapeseed honey, crystallizes quickly, while honey with a predominance of fructose, acacia honey, fireweed honey, remains liquid for years. The second important enzyme is diastase amylase. Its role is to break down starches and dextrins, which may be present in nectar, especially honeydew, into simpler sugars. Diastase activity is one of the most important indicators of the naturalness and maturity of honey, as well as traces of possible heating, as this enzyme is heat-sensitive and quickly degrades at high temperatures. The third, no less important process is the action of the enzyme glucose oxidase. When honey is placed in cells and its moisture content decreases, this enzyme begins to slowly work, oxidizing some of the glucose to form gluconic acid and hydrogen peroxide. It is this step that gives honey its unique bactericidal and preservative properties. Gluconic acid is responsible for honey's consistently acidic reaction, pH 3.5 to 4.5, which creates an environment unfavorable for the development of the vast majority of bacteria and microorganisms. Hydrogen peroxide, on the other hand, is a natural antiseptic that further inhibits microbial growth. Interestingly, the process of hydrogen peroxide formation is activated when honey is diluted, for example, in a wound or in the digestive tract, which explains its traditional use in folk medicine. Parallel to the enzymatic reactions, the physical process of water evaporation occurs. By actively ventilating the hive, bees reduce the humidity from the initial 40–80% to 17–20%. This low humidity, combined with the high osmotic activity of the concentrated sugar solution, which literally pulls moisture from the microbial cells, makes honey an even more inhospitable environment for spoilage. The result of this cascade of reactions is the creation of a substance in which fermentation and decomposition processes are virtually impossible. The sugar composition, acidity, low humidity, and the presence of natural antiseptics make honey one of the most stable natural foods on Earth. This stability is the main result of the chemical transformation that the bee colony undergoes, providing itself with a waste-free and everlasting food supply. The bee colony's work ends with the sealing of the honeycombs with wax caps. At this stage, the beekeeper becomes involved, whose job it is to carefully remove any excess honey without harming the colony or affecting the quality of the honey. This process involves a precise sequence of technological steps that require skill, knowledge, and careful handling. The first and most crucial step is determining the honey's readiness and selecting the frames. An experienced beekeeper will never extract unsealed honey, as its moisture content exceeds 20%, and such a product can quickly ferment. A sign of maturity is tightly sealed, crusted honeycombs, where each cell is covered with a smooth wax cap. The beekeeper inspects the hive, locates frames with fully sealed honey, most often located along the edges of the brood nest or in the upper bodies of a horizontal or multi-body hive, and carefully removes them after brushing off the bees with a soft brush or shaking them off. A smoker can be used to calm the bees. The smoke triggers their instinctive hoarding response, causing them to fill their crops with honey, making them less aggressive. The next step is uncapping. This is the physical removal of the wax caps to free access to the honey. There are two main methods. The traditional method is to use a special apiary knife heated in hot water, which carefully cuts off a thin layer of cappings. A more modern and gentler tool is an uncapping fork, or roller, which opens the cells rather than cuts them off, minimizing wax and honey loss. The cappings collected during this process are a valuable product in their own right, rich in enzymes and propolis. The main operation, honey extraction, is performed using a honey extractor. This is a centrifuge, usually radial or cordal, in which uncapped frames are mounted on special cassettes. As the rotor rotates, centrifugal force is created, and the heavy honey is sprayed out of the cells onto the walls of the extractor, from where it drains to the bottom. The process is carried out carefully at low speeds to avoid damaging the delicate wax combs, which, after extraction, are returned to the bees for reuse. It is critical that all equipment that comes into contact with the honey be made of non-reactive and non-oxidizing materials, food-grade stainless steel, coated aluminum, or wood. After extraction, the honey is a thick liquid containing impurities, tiny particles of wax, propolis, air bubbles, and sometimes bee bodies. Therefore, the next step is filtration and settling. First, the honey is passed through a coarse filter, often made of stainless steel mesh, which retains large particles. Then it is allowed to settle in special containers, sedimentation tanks, or cages for several days. During this time, heavier impurities sink to the bottom while fine wax particles and air bubbles float to the surface. This top layer is carefully removed. Some beekeepers practice additional fine filtration, but excessive clarification deprives the honey of some valuable components such as pollen. The final stage is bottling and storage. The mature, filtered honey is poured into clean, dry containers. A dark glass jar is considered ideal as it protects the product from the damaging effects of direct sunlight. Ceramic vessels or wooden barrels made of linden, beech, or alder are also suitable. Plastic is not the best choice for long-term storage. The correct storage conditions are simple: cool, dark, and dry. A temperature of around 10 to 20 degrees in a dry room allows honey to retain all its properties for years. Thus, the beekeeper's role is that of a careful technologist and guide, completing the bee's work, transforming honey from the colony's biological reserve into a product ready for human consumption, all the while preserving all the complexity and benefits created by nature. This complex and energy-intensive process of honey production is not a whim of nature, but a brilliant survival strategy honed over millions of years of evolution. For a bee colony, honey is not a delicacy, but a vital strategic energy reserve, enabling it to survive long periods when collecting nectar and pollen is impossible. Chief among these periods is the cold season. In winter, when temperatures drop below 8 degrees Celsius, bees cannot fly, and there are no flowering plants. To survive, the colony forms a dense cluster, maintaining a temperature of around 15 to 20 degrees through muscle tremors and energy consumption. The source of this energy is the honey stored in the honeycomb. The bees use it sparingly, slowly processing it and releasing the necessary heat. Without sufficient reserves of high-quality mature honey, the colony is doomed to die from starvation and hypothermia. Furthermore, honey reserves are critical during adverse weather conditions during the warm season, during prolonged rains, droughts, or early frosts when foraging flights are impossible for several days or weeks. The ability to produce and store concentrated, non-perishable food gave honeybees a huge evolutionary advantage over other insects, allowing them to colonize regions with pronounced seasonality and harsh winters. It was honey as a universal preserved food that made possible the existence of huge colonies of thousands, which, unlike solitary bees, can plan their existence months in advance. There's an important distinction between honey for bees and honey for humans. A responsible beekeeper understands that they don't take honey from the bees, but only remove any surplus the colony has produced beyond what is necessary for its survival and development. The fundamental principle of ethical beekeeping is to always leave a sufficient supply of food in the hive for winter and adverse weather. This supply is calculated based on the colony's strength, climate zone, and the duration of the hive's wintering period. Only after the colony's needs are fully met can the remaining honey be harvested. Thus, beekeeping is essentially a symbiotic relationship. We create optimal living conditions for the bees, protected hives, treatment for illness, supplemental feeding during food shortages, and in return we receive a share of the fruits of their labor. The honey we buy is the result of this ancient partnership where the well-being of the bees comes first. Ultimately, all the titanic work of collecting nectar, fermenting it, and storing it has one main goal: to ensure the continuity of life of the most complex social organism, the bee colony, from one generation to the next, from season to season. The journey from a drop of nectar on a flower to golden honey in a jar is a grand story, intertwining biology, chemistry, and complex social organization. We have learned that honey is not simply a product collected by bees, but rather created through a multi-stage production process comparable to a high-tech factory, where each participant plays a distinct role. Beginning with the exploration and extraction of raw materials, bees consistently carry out enzymatic processing, the removal of excess moisture, and the final hermetically sealed packaging in perfect wax containers. Each stage, from the scout's dance to the sealing of the cell, is aimed at achieving a single goal: converting perishable sap into concentrated, stable, and energy-rich food capable of sustaining an entire colony for many months. The main conclusion we can draw is that honey is the quintessence of the collective labor and wisdom of the bee community. It is a product that encompasses thousands of kilometers of flight, millions of visited flowers, and the flawless operation of a biological conveyor belt. Understanding this complexity engenders a deep respect for both the bees themselves and the work of the beekeeper, who acts as a careful guide, completing this process and sharing its fruits with us. Therefore, while savoring the taste of honey, it's worth remembering that every spoonful is more than just a sweet treat. It's the embodiment of ingenious natural engineering, a survival strategy, and a symbol of the incredible harmony that reigns within the hive. By choosing genuine, mature honey from responsible producers, we not only receive a product unique in its properties, but also maintain this delicate balance between humans and nature.
That's all for today's video. Don't forget to like and subscribe to the channel so you don't miss any videos. See you soon.